Hey there, third grade teachers, Mr. Height, joining you to go over this new lab that we have for third grade. If you were a third grade teacher in the past, you remember we did a lab called Plant Parts, Glorious Plants, different titles. And we would go over all the different parts and the functions of the plant. What we're going to do this year is really focus in on the leaves of the plant, specifically how plants make their own food and the different materials they use to make their own food. And so the lab demonstrations will be looking at evidence that plants are making their own food. So I just kind of wanted to show a couple of the materials you might need and just kind of run through some of those days. So I'm going to go ahead and pull up the guide here, and there are student pages obviously for this as well, which we'll take a look at. So here is our lab guide and our two standards. We're definitely looking at the roles of the different parts because those are still very important, especially distinguishing between roots and leaves, because students still think that roots actually provide food for the plants, and we know that's not true even in fifth grade. That's why we're focusing on the leaves here, is even in fifth grade they still struggle with that food production piece of plants. So we thought maybe let's make a lab that really focuses on food production and the leaf of the plant. So that's what we're doing here. We are going to talk about photosynthesis. Yes, we can do that. Totally perfect to talk about that. That is the process plants use to make their own food. Do they need to get into the cellular level or the chemical formula for photosynthesis? No. But we can tell students that plants use water, sunlight, carbon dioxide to make their own food, and that process is called photosynthesis. So that's totally cool. But they don't need to get into like chemical formulas and the breakdown of the atoms and things like that. No. A little teacher background for you. And I did put a note in there that yes, we are going to use that word photosynthesis. Totally cool to use that as well. Materials there, kind of light materials. I'll show you a couple of the different kits and things we will use for that. So day one, they will need some Ziploc baggies or a little container because they are going to go out on campus and try to locate a leaf, a root, a stem, a flower, so parts of plants, and identify their function. And also in day one, setting up the celery experiment. We left that in there, so let me show you a quick picture. So we had this in the past, but what we're going to do is on that first day of the lab is go ahead and set it up. So the lab manager might already have this. So we want some celery stalks in water with some food coloring, and blue or red are usually the really good colors, and you just need one of these stalks per table group if you want to do that. We'll start it on day one, and then day 5 students will actually get a chance to dissect and open up the stalks and the leaf of the celery. So we just want to see how water and different materials are transported through the plant, and especially focusing on the leaf. Absolutely. So they'll start that on Monday and ask some prediction questions. And then students every day that come in the lab, they can kind of look at their stalks and see what is happening, make some quick observations, but no, just on that last day, they will actually complete a dissection of the celery stalk. So that's a lot of fun. So we did leave that one in there. It's a good observation, good hands-on demonstration there, especially dissecting. Very cool. So that's what day one is. They will go out and do some collecting. We will start off with a probe. So in their student pages, there's an apple tree probe, excellent probe, because it's just having students think about, okay, where is the food made? So you get to really see some misconceptions. I'll just say historically, they think the roots. They really think that's where everything's absorbed, water, and then food goes in through the roots. That's what they think happens. And sometimes adults do that too. We think that, and we say that. We even say plant food, fertilizer, which is not right. It's not right. It's nutrients and minerals which help the plant, but they don't feed the plant. They'll help the plant grow and be more efficient, but it's not actually providing food for the plant that is made in the leaves. So that first day, they'll go out and take a container, a sandwich baggie, whatever, and try to find some parts of plants, and they record that in their lab journal provided. We'll take a look at that pretty soon here and do a little quick sketch. They could collect them and bring them back in the lab if you want to do that. That's totally cool, and then they can do their observations, have hand lenses always, especially this week. Great to have hand lenses available so the students can really observe what is going on. So we want them to do that. If you want to take a step further, if it's weather or whatever, if you want to have plant parts already available in the lab, that would be totally cool too. If you're not able to do a campus walk. So you can set that up, which is best for your class, for sure. And of course, we got a couple of songs there, one by a very good performer. You know him very well. So that song is in there, along with some other songs as well. And then day 2, this is where we actually get to the ID kits. Of course, when they come in, let them kind of look at their celery stalks just real quick. Hey, do you see anything happening with the celery stalks? So let them take a look at that. And then we will get into the first part of this, which is they will use the ID kit, the leaf identification kit. Lots of schools should have this, and we just sent out a refresh on this, so you might have another kit. And in these kits, lots of, there's a little handout. Don't really refer to that, and there is a nice little ID tree finder, which is pretty cool to look through. But then there are all sorts of leaf samples. So what I usually do is have the students do a little quick sort. How would they sort these different leaves? And then there's actual in the guide here and actual in the student pages, we want them to actually pick out some specific leaves to do some comparisons. Getting to the point is, why do leaves come in different shapes and sizes? Why is it necessary? Why do some plants have massive leaves? Why do some plants have very small leaves and different shaped leaves? Different like skinny leaves and things like that. And I believe I even have some conifer leaves. So very, very small leaves. So having students understand why leaves come in different shapes and sizes, all having to do with obviously where they're grown, so their environment, and the amount of sunlight. So some plants that grow in low sunlight areas might have to have very large leaves. Other plants that grow massive might need small leaves. They might have a lot, but they have a lot of small leaves. Plus, they can store a lot of food. Some plants they don't have a lot of food storage, so they have to constantly go through photosynthesis. So we want them to understand that leaves do come in different sizes and shapes because of where they grow, environment, available sunlight. The soil could be also a part of that as well, and food storage. So that's what we have there. Then it moves into, go back over here. So they'll do a little leaf sort, and there's some questions obviously in the student journal there that we provide. The next thing is we want the students to collect some green leaves. And notice I put non-waxy leaves because we want to look for evidence of photosynthesis. This can be done ahead of time, so just get a bunch of nice green leaves you'll see here, I'll show you in a second. And a sandwich bag is fine, or even a gallon bag is fine. And make sure the bag is sealed and that there's air in the bag. And then I would tape the sealed bags to a window where it can get some sunlight. And then the students will predict what they see there. Okay, so I have just a little example. So I went out and got some green leaves, got a gallon bag, but you can use sandwich bags, it'll work fine. Just make sure that there is air inside the bag, and then tape that to a window that has some sunlight and leave that overnight. They'll predict. And if everything works the way it should, you know, it is science, but we're looking for evidence of photosynthesis. As we know, plants, while they're going through photosynthesis, give off a couple things: oxygen, but also water vapor. We know that photosynthesis is happening if we start to see some moisture inside the bag. We know that that the leaves are going through photosynthesis, so that is evidence of photosynthesis. So they will set that up, do some predicting, and you just need one bag per small group. That's totally fine. So depending on how many small groups you have, and they'll put that on the windowsill somewhere that has got sunlight going through there. Let that set overnight into the next day. Hopefully you should start seeing some things happening. If not, you could even leave it up a couple more days. It just depends on the type of leaf that is in there and the air and available sunlight. So and you should see some moisture start to occur in the bag, which is pretty cool. So that is one of the hands-on activities for the lab. And also provide some exit questions there too. And I even put a little thing, what should happen. So again, those bags, the leaf in the bags, we're looking for evidence of photosynthesis. So plants release water back into the air, evaporation, as water is used for photosynthesis. So we know plants are making their own food if we see some moisture happening in the bag. So again, next day, day 3, they will come in, look at their celery stalks, and they will actually go into looking at their leaf bags, so recording their observations. And you can leave them up a little bit longer. If nothing's happening, okay, we'll come back again, but should start seeing some things happening. Make sure students have hand lenses, all that good stuff. Then they're going to set up another investigation. And this one again is very simple. What they need is a clear plastic cup and a nice large leaf, not a huge leaf, and a paper clip, a large paper clip. And I'll show you that in a second, per small group. So again, we're going to look for evidence of photosynthesis. So we know that plants release oxygen back into the air as a result of photosynthesis. So what I have here is I have a nice leaf, and I'm going to submerge it. I have a paper clip in there to make sure that the leaf stays submerged. So the leaf goes in here in the water. Okay. And the interesting thing is I've had it in here for a little bit, and you're already seeing some interesting things. I've had this sitting in here for a while, and you want to also keep it in a sunny location. And if you saw what I'm seeing there, you are seeing some bubbles, so some air bubbles. So some evidence of photosynthesis is plants make oxygen. That's a byproduct of photosynthesis, releasing oxygen back in the air, which is awesome for animals. Yay! And we can see that through the little bubbles there that form in the cup. So that's what we're going to be looking for. Obviously, they'll do some predictions as what they might see the next day, but they'll set that up. So lots of observations this week with the celery, the ID kit, doing some things with the ID kit, doing our leaves in a bag, and then a leaf in a cup. So lots of little demonstrations as we go through this new lab, but I like it because it's nice and focused. really focusing in on one major part of the plant. Okay, going into day 4, they will then come back to their cup and take a look at what's going down, or what's going on, what's going down and what's going on. Also have them look at their celery stalks again. We're not opening them up yet. They just can be on the side of the lab on the 3rd grade table, taking a look at those celery stalks, see what's going on there, making some observations. Let them know on day 4 that on day 5 they will begin to dissect and open those up. So for this day 4, we're going to take our leaf. Hopefully we've seen some bubbles forming, either on the leaf or maybe you might see some on the side of the cup, which is pretty cool. And as we know, leaf is making oxygen, releasing that back into the air. What we'll do here then, we'll take the leaf out, and you'll need a paper towel, and we'll let students do a leaf squeeze. So they'll take a pencil or some other object to kind of press down on leaf and kind of roll it and squeeze the leaf, and they'll probably see some water, but they also might see some green substance, because that's what we want to use, green leaves. They might see some green substance, and you can tell the students that this is chlorophyll, and it gives plants that green look, that green color, and you can let them know that chlorophyll is what makes photosynthesis happen, with that chemical inside the leaf. So they can actually see some of that chlorophyll, hopefully, on their paper towel when they do the leaf squeeze. So we're going to let them squeeze the leaf, get the water and the chlorophyll hopefully out on a regular paper towel. I wouldn't use the brown paper towels. I would use like the white absorbent paper towels. It's a good thing to do. We also have a PowerPoint. I would definitely do the PowerPoint. It's a good content building PowerPoint called Food Photosynthesis. It's on Canvas, so I would go through that and have the students think about those different slides, and there's a couple questions that go along with the PowerPoint. We definitely want to make sure we ask questions when we're using digital media there, for sure. Okay, and then we go into our last day, day 5. This is where they actually get to dissect their celery stalks. So you'll take our celery stalks out of the water. Pull that up, our picture here. And make sure each table group gets a celery stalk with the leaf. That's very important when you do buy lab managers. When you get your celery stalks, make sure they do have leaves on them. Very important that you have that available. And then you have some paper towels. Let the students break open the celery stalks. They'll be able to see the tubes and different ways the water gets around, but also asking the questions besides water, what other ingredients might travel through the plant and through the leaf. Hopefully they'll say food for the plant. The sugar also is transported throughout the plant and the leaf, so it gives them opportunity to kind of look inside the structure of the stem and the leaves. And using that water, you can see how it goes through all the parts of the plant. So they really like doing the dissection there. And of course there's a couple of questions that involve that dissection as well that they can ask. When you wrap that up, I would go back to the apple tree probe and re-answer that probe. Hopefully they will answer that food is made in the leaf. So hopefully their thinking changes if they chose the wrong answer, for sure. So we want to make sure that they're doing that. Move this over here a little bit. And then there are some additional resources. So if you do have some extra time, there's a couple different other photosynthesis songs. There's actually a sea palms tutorial, even some textbook pages, Science Source. The digital lab is really cool for using sunlight and making food for the plant. That's a good activity as well if you have extra time. And also on that last day, make sure you do your food from photosynthesis post-test, so make sure your students do that as well. Let me show you the student pages, just so you know what they're recording in their student pages. So I'll bring that up. So here is our first day with our tree probe, apple tree probe, our vocabulary, I can statements, essential question. And that would be a daily thing, having them always go back to the I can statements and things like that. Apple tree probe, and they can do their answer there. I like this because it actually has them what they're thinking, and then they have to choose a different thinking. So if I chose Molly, then I want to choose maybe Jared and say why I think Jared is wrong and Molly's right. So they will do that. 2 answers they have to provide. And then part 2 is with their group going out and collecting some plant parts. So getting a stem, having them understand what the stem is, and doing a quick sketch of each of these plant parts. Hopefully helping them understand a little bit more about those plant parts. Again, we want them to understand that the leaf makes the food and the others don't. And we know that they're going to really always gravitate to the roots. So once they understand the role of the leaf, they'll hopefully start to separate that, oh, the roots just absorb water and nutrients, which are very important, but that's not food. And then day 2, go leafing. This is where they do the ID kit. So they'll do some sorting and do a couple of different questions here for different shapes and sizes of leaves. Then they'll do the leaf hunt. This is where they're going to get their leaf in a bag, their 6 green leaves, put them in a sandwich bag, and then tape it to the window with some sunlight. And they're going to predict what they think is going to happen, and when they observe it the next day. And then for that next day is right there. So once they observe the next day, they should do a labeled illustration of what they saw happening. And they can come back to that if you decide to leave those bags up a couple more days. They could always come back and add to this drawing, of course. Very cool. And then we have our leaf in a cup demonstration. So they will draw a picture and a predict. If they submerge their leaf in a cup, they'll draw what they think is going to happen, also write what they think they will observe. And then obviously on day 4, they'll come back to it. Hopefully they'll show that some bubbles have formed and what they think is happening. Why are they seeing bubbles there? And then the last thing we have in the student pages is the celery dissection. They'll draw and label the celery, hopefully the parts, and then based on their observations, why do you think water is an important need for plants? So that is for day 5. So that is our student pages. And if you have any other questions, just shoot me an email. I'd be happy to answer any of those, but hopefully this video helps you when you begin your planning and your preparation for this lab, and getting your students excited about learning about how plants make food. So thank you all for joining me, and I'll see you real soon. Have a good day.